Autism brain signature most pronounced in sensory areas. Gene expression differences linked to autism appear throughout the cerebral cortex, but are most pronounced in sensory parts of the brain, such as the visual cortex. But in autism, more genes are dysregulated as you go to the back of the brain, and that kind of gives us some clues as to the origin, potentially developmentally, of when these changes are happening. But these signatures are based on small samples limited to association areas in the temporal and frontal lobes, which control higher-order functions such as cognition, language and social behavior. I think most people wouldn't suspect that these regions have the biggest vulnerability. Gandel and his colleagues analyzed 725 post-mortem brain samples from 49 autistic people and 54 non-autistic controls. The brains of autistic people showed dysregulation in 4,223 genes and 9,474 gene isoforms, RNAs from the same part of the genome that often have different protein coding sequences. Their results allow us a glimpse of molecular mechanisms at play. The dysregulated genes aren't randomly spread across different cellular processes either. They're in very specific cell types, particularly neurons, oligodendrocytes and microglia. Says study investigator Daniel Vishvant, distinguished professor of neurology, psychiatry and human genetics at the University of California, Los Angeles. Across the entire cortex, 13 gene modules, groups of genes that exhibit similar expression patterns, had regional variations. All of these modules followed an anterior to posterior gradient, meaning they were most dysregulated toward the back of the brain. In BA17, dysregulated genes include SOX4 and SOX11, transcription factors to promote neuronal differentiation, which encodes a sodium channel for cellular signaling. In this region alone, more than 3,200 genes were differentially expressed compared with non-autistic brain tissue. One gene, called DTV4, encodes a transcription factor that helps dendrites to develop and connect with neurons. Genes that are regulated by ETV4 were also reduced in the BA17 region in autism brain samples. That analysis showed that autistic people have slightly more astrocytes and fewer excitatory neurons, but the differences in cell type numbers weren't dramatic enough to explain the findings. What this study is asking: at the single-cell level, are there actually changes in molecular pathways and signaling that are going on within cells? Gandel and Geschwind are now testing various hypotheses to explain their results. They are expanding their analyses to include the thalamus. If correct, that this is reflecting sensory input into the cortex, then we would expect that the thalamus, which is the major relay station of sensory information in the brain, would show a similar pattern of dysregulated genes. But there's much more to explore outside of these sensory regions too. Non-cortical parts of the brain, including motor regions in the cerebellum and brainstem, have similarly been implicated in autism. Remember, knowledge is power.